Okay, let's dive right into this. Did you know your body produces enough saliva in a single year to literally fill 2 entire bathtubs? Yeah, it's wild. But have you ever stopped to wonder what actually happens after you swallow a bite of your favorite food? How does your body take something like a massive sandwich and turn it into the very energy that keeps your heart beating and your brain working? Well, welcome to this explainer. Today, especially for you O-level biology students out there, we are going to completely demystify human digestion. We're going to break down this incredible journey from a single bite of food all the way down to microscopic energy.
So here's our chronological roadmap for today. We'll start with beyond the bite, track the mouth-to-stomach journey, dive into the small intestine, see digestion in action, and finish up with common mistakes and a recap.
All right, Section 1, Beyond the Bite.
Think about eating a giant sandwich. That whole sandwich is, well, it's just way too big and complex for your body's cells to actually absorb and use. Digestion is simply the process of breaking down those complex food molecules into simpler absorbable units. We're talking glucose, amino acids, and fatty acids. Your body literally has to dismantle the macro food into these specific micro units just to survive.
Now, what's really interesting here is that there are two ways this dismantling happens, and they happen simultaneously. First, you've got physical digestion. It's kind of like physically tearing a piece of paper into smaller pieces. This mechanical breakdown increases the overall surface area. But at the exact same time, we have chemical digestion. And this isn't just changing the shape of the food. These are enzymes actively breaking the invisible chemical bonds that hold the molecules together. Basically, physical digestion preps the food, giving those chemical enzymes a much larger surface area to attack.
Moving on to Section 2, Mouth-to-Stomach Journey.
So, the alimentary canal is essentially this one long continuous tube running all the way from the mouth down to the anus. We're going to track a bite of food right through this exact anatomical sequence, starting right at the top.
Our journey kicks off in the mouth. And while the macro physical chewing is happening, your salivary glands are pumping in all that saliva. This contains water to moisten the food and mucus to bind it together. But crucially, it contains salivary amylase. So while you physically chew on the outside, on a microscopic level, this enzyme is instantly starting the chemical digestion of starch.
Once that chewed food turns into a soft ball, which we call a bolus, it drops down into the esophagus. Here, these really powerful, wave-like muscle contractions forcefully push the food down the tube. This is called peristalsis, and you can visualize the directional force pushing it downward. Note that absolutely no digestion happens in this step. It's purely a transportation mechanism to get the food safely to the stomach.
And boom, we land in the stomach. Now the stomach is a pretty violent environment. It's literally churning and tossing the food around, violently mixing it with gastric juice. This juice is a super potent mix of hydrochloric acid, pepsinogen, and the active enzyme pepsin. It's this highly acidic, highly active mixing bowl where both physical and chemical breakdown are just happening at full force.
Let's walk through exactly how this builds step by step. First, that gastric juice fully mixes with the food. Next, our enzyme pepsin locks right onto its specific substrate, the protein bonds. Finally, those large proteins are transformed into smaller, way more manageable chains called polypeptides. So you have the enzyme, the substrate, and the product. Beautifully organized.
All right, Section 3, the small intestine. Now, this next part of the process relies on some incredible teamwork. Associated glands, meaning the liver, the pancreas, and the intestinal walls, they all secrete these powerful juices directly into the small intestine. And packed inside these secretions are some real heavy-hitting enzymes, pancreatic amylase for carbohydrates and trypsin for continuing that protein breakdown.
Okay, pay super close attention here, because this is a major exam trap. Enzymatic action, like what trypsin does, chemically breaks bonds. But bile does not break chemical bonds. Instead, bile acts as a physical preparer. It takes these giant, clumpy fat drops and physically breaks them apart into tiny droplets. That's called emulsification. Emulsification is a physical process, not chemical. Definitely don't let the exam trick you on that one.
Let's move to Section 4, Digestion in Action.
Let's put this all together and track a specific carbohydrate through the flowchart. Step 1. We start in the mouth, where salivary amylase attacks complex starch, turning it into maltose. Step 2. The stomach is actually bypassed for carbs, so we move right to the small intestine, where pancreatic amylase finishes breaking down any remaining starch. And finally, Step 3. The enzyme maltase steps in and breaks that maltose down into our final absorbable unit, glucose.
Now, take a look at this matrix. This is seriously your ultimate study tool. You need to memorize the exact mappings of location, enzyme, substrate, and product across the entire digestive tract. If you can recreate this grid from memory, stamping every core variable directly onto its corresponding row, you're going to be in incredible shape for your test.
Finally, Section 5, Common Mistakes and Recap.
Let's just quickly clear up two massive misconceptions. First up, the idea that digestion only occurs in the stomach. Absolutely false. As we saw, chemical breakdown starts immediately in the mouth with your saliva. Second, the idea that emulsification is chemical. False again. Remember, it's purely a physical process that just separates fats to increase the surface area for the actual chemical enzymes to do their job later.
So to recap, digestion is this beautiful, highly coordinated effort. It relies on a continuous tube working right alongside associated glands. It demands physical surface area expansion through chewing, churning, and emulsification, which perfectly sets the stage for those chemical enzymes to come in and break the molecular bonds. And that is exactly how you turn a simple sandwich into usable microscopic energy.
I'm going to leave you with this final, kind of provocative question to test yourself before you go. If you ate a plain starch cracker right now, exactly how many different organs and enzymes would it physically pass through before becoming usable energy? Really try to mentally trace that chronological pathway for yourself. Thank you so much for joining me for this explainer, and best of luck on your exams.